Okay, let's start. Number 1. The Amarna Egyptians. You're in the Valley of the Kings in 1907. Edward Ayrton, excavating under the patronage of Theodore Davis, opens Tomb KV55, a small corridor chamber sealed with the cartouche of Tutankhamun, but packed with material bearing the erased name Akhenaten the Pharaoh, whose successors spent 2 decades systematically cutting his image and name from every monument in Egypt. Inside, against the western wall, is a coffin. The gold portrait face has been methodically pried off. The cartouches have been hacked out. The mummy inside has been desecrated post-interment, the arms repositioned, and the identifying marks removed. Anatomist Grafton Elliott Smith receives the skull in Cairo and measures it. He has catalogued thousands of Egyptian crania. He notes in his 1910 report that the posterior cranial vault extends beyond any morphological range in his reference collection and does not correspond to any identified pathological condition in human bone literature. The mainstream explanation operates on 2 tracks. The first is artistic convention. The Amarna period's distinctive depiction of the royal family elongated skulls, prominent abdomens, wide hips, narrow chinsies understood as a stylistic innovation chosen by court artists, not a naturalistic portrait. The second track is genetics. KV55's unusual skull represents an unspecified familial condition, possibly Marfan syndrome, possibly Antley-Bixler syndrome, possibly familial craniosynostosis, that affected Akhenaten and produced the observable anomalies. Here's what makes physical anthropologists uncomfortable. Amarna art is the most naturalistic in all of Egyptian art history, and the morphology is not confined to Akhenaten. For 2,000 years before the Amarna period, and for centuries after it, Egyptian royal portraiture idealized the pharaoh as young, muscular, symmetrical, and beautiful. Amarna art depicts Akhenaten as unmistakably strange. A protruding abdomen, gynecomastoid chest, wide hips, elongated limbs, and a skull that rises and extends several inches further in the posterior superior direction than any normal human cranium. This is not flattery. No ruler commissions court art portraying him as physically abnormal without a reason. The working assumption in mainstream Egyptology is that Akhenaten deliberately introduced a new religious aesthetic. The problem is that the same skull shape appears on every member of his immediate family, including infants, in scenes that carry every other marker of domestic naturalism. Of the 6 royal daughters, every individual depicted on the limestone stelae and wall carvings of Amarna carries the same distinctive posterior cranial extension. Barry Kemp of Cambridge University and the Amarna Archaeological Project documented skeletal material from 2 child burials excavated at the city site of Amarna itself. Both skulls show the same elongated posterior vault. No binding artifacts were recovered from either burial context. In 2010, Zahi Hawass and colleagues published a DNA study of royal mummies from this period in JAMA. The analysis confirmed that KV55 is the biological father of Tutankhamun. It documented a cluster of familial conditions, including malaria and bone disease, running through the same lineage across multiple generations. The skull morphology of KV55 was noted, classified under hereditary pathology in the broadest possible language, and not resolved. No published study has identified a hereditary condition that simultaneously produces elongated posterior crania, abdominal prominence, gynecomastoid chest features, and limb proportion anomalies across multiple individuals in the same nuclear family without leaving a trace in any descendant population. Bob Breyer at Long Island University has proposed familial glucocorticoid deficiency as a candidate explanation. The diagnosis is contested. Erwin Braverman at Yale proposed a different endocrine disorder from the same artistic evidence. The 2 diagnoses do not agree. Neither accounts for the full morphological cluster. The features disappeared completely. Not gradually, not through admixture completely. The successor dynasty under Horemheb produced no skulls, no art, and no children displaying any of these characteristics. The Amarna royal line produced an entire family with a consistent and specific skeletal anomaly, and when that family ended, the anomaly ended with it. What genetic condition produces identical elongated posterior crania in every depicted member of a royal family across 3 generations? What happened to the descendants? Why does the anomaly vanish from the art and the bone record simultaneously, at the precise moment the dynasty is forcibly erased? And who decided, before sealing Tomb KV55, that this particular face needed to be destroyed. 7,000 miles southwest, another people buried their royalty in coastal cliff caves above the Pacific and left behind skulls that no model has explained in a century. Number 2. The Paracas people. You're on a wind-scoured peninsula on the southern coast of Peru in 1928. Julio Cesar Tello, the father of Peruvian archaeology, is directing the excavation of burial caves cut into the coastal cliffs above the Pacific. What comes out of those caves will stay with him for the rest of his career. Not the textiles, extraordinary as they are. Not the ceramics or the burial goods. What stays with him is the skulls. Over the following months, Tello's team recovers 429 mummified individuals. Nearly all of them carry the same skull, elongated dramatically backward from the crown, extending several inches beyond any normal human cranial range, wrapped in the same funerary cloth, buried in the same posture. 429 of them. The mainstream explanation is cranial binding. Wrapping an infant's skull with boards or cloth during the first years of life, when the skull plates are still mobile, gradually reshapes the growing bone into a different configuration. The practice is documented across multiple cultures on multiple continents. The Paracas people practiced it intentionally. The elongated skulls are cultural markers of status. They are reshaped normal human skulls. Here is what makes anatomists uncomfortable. Head binding does not add volume. It redistributes existing space. It compresses the skull laterally or vertically, depending on the method, without generating new internal capacity. The internal cranial volume stays essentially the same through binding. The Paracas skulls do not stay the same. Detailed measurements of multiple Paracas specimens document internal cranial volumes ranging from 2,200 to 2,500 cubic centimeters. The average modern human cranial capacity is 1,350 cubic centimeters. The average Andean skull from the same archaeological period sits near 1,200 cubic centimeters. The Paracas skulls contain between 60 and 100% more internal volume than the surrounding population. Binding produces a different shape. It cannot produce 60% more brain space. The physics do not allow it. There is a second anomaly that no form of binding can explain. The human skull has two parietal bones joined along the top of the head at the sagittal suture, one of the most consistent structural features in human anatomy across all populations and all periods. Multiple Paracas skulls examined by researcher Brian Forrester, beginning in 2009, show a single continuous parietal plate where two bones and a suture should appear. No seam. One piece of bone running the length of the skull's crown. This is not a feature produced by deformation. This is a different bone architecture. In 2014, Forrester submitted samples from 18 Paracas skulls to 3 independent genetic laboratories for mitochondrial DNA analysis, testing for haplogroups, the genetic markers that identify which ancient population an individual descended from. The results from all 3 laboratories were consistent with each other. The haplogroups identified did not match any known Native American population. They did not match European populations. They did not match any Asian population along the established Bering Land Bridge migration corridor. One of the 3 laboratories reported sequences that did not correspond to any known human mitochondrial haplogroup in the database at the time of testing. The 2014 results have sat in a private report for over a decade. No major university anthropology department has commissioned independent replication. No government archaeological body has funded a peer-reviewed follow-up study. The Paracas skulls, hundreds of them, sit in museum collections in Peru and in private institutions worldwide. The largest collection is at a small museum in Paracas itself, where the skulls can be photographed but not sampled. The question hangs open over the bones, and nothing has moved toward it. Where did the Paracas people come from? What population has 60% more cranial volume than the people buried beside them, with a bone architecture that differs at the structural level, producing mitochondrial DNA that fits no migration model on record? When the culture ended around 200 CE, where did they go? And who, in institution after institution, decided that the 2014 results did not warrant a follow-up? 429 skulls. No answers from any of them. On the other side of the world, in a desert in western China, another population left behind bodies that should not be there, and the government that found them has spent decades managing what happened next. Number 3. The Tarim Basin mummies. You're in a laboratory at the University of Pennsylvania in 1993. Victor Mayer, professor of Chinese language and literature, is looking at photographs taken by Chinese geological survey teams from burial sites in the Tarim Basin, a vast inland depression in western China's Xinjiang region, so hyper-arid it receives less than an inch of rainfall annually. The photographs show mummies, not deliberately mummified in any formal sense, but naturally preserved by extreme desiccation and salt in a condition rivaling anything found in Egypt, and they are visibly not Chinese. The mainstream explanation is Bronze Age migration. During the 2nd millennium BCE, populations from the Pontic-Caspian steppe, the vast grassland stretching north of the Caucasus from Ukraine to Kazakhstan, expanded eastward into Central Asia. The Tarim Basin mummies represent an early branch of this expansion, preserved by accident of climate. The Indo-European linguistic group spread the same way. The mummies are interesting and well preserved, but not structurally anomalous. They are exactly where expanded migration models predict a steppe-derived population would appear. Here's what makes population geneticists uncomfortable. The Tarim Basin mummies did not come from the steppe. The Lulan beauty, recovered by Chinese archaeologist Wang Binghua from the Koraghul Cemetery east of Lop, Nor in 1980, dates to approximately 1800 BCE. She is 5 feet 2 inches tall, with reddish-brown hair and facial proportions consistent with Western Eurasian populations. She is buried with woven wool textiles, not documented in Chinese archaeological records until more than 1,000 years later. The Churchen Man, recovered from Zaganluk in 1985 and dated to approximately 1000 BCE, is 6 feet 6 inches tall, with auburn hair and a full beard. He wears a red and white wool tunic and plaid trousers, whose weave matches textile types from Celtic period burials in Central Europe. He died in what is now Western China. Victor Mayer arranged for DNA analysis in 1993. Results confirmed Western Eurasian ancestry haplogroup R1A1 in the male line, fitting the steppe migration model. Then the 2021 ancient DNA study by Chumwon Jiang and colleagues, published in Nature, changed everything. The Tarim Basin people were not migrants from the steppe. They were a genetic isolate. Their ancestry derived from an ancient North Eurasian population present across northern Eurasia during the Pleistocene, but without confirmed surviving modern descendants in any isolated form. The steppe populations who would later expand across Eurasia were not their ancestors. The Tarim Basin people were older, genetically separate, and arrived from an unknown direction before the Yamnaya expansion began. The 2021 paper found no detectable genetic contribution from steppe, South Asian, or East Asian populations. In a region that subsequent history would see traversed by Silk Road trade for 2,000 years, the Bronze Age Tarim Basin population remained genetically unmixed. That degree of isolation requires either deliberate endogamy of an extreme kind or a population dynamic that has not been explained. The Chinese government restricted external access to the mummies and their associated materials beginning in the 1990s. The most complete collection is displayed at the Xinjiang Museum in Urumqi. External research sampling requests have encountered institutional delays running for decades. Victor Mayer has described the political context directly in published interviews. A genetically Western population occupying Xinjiang for 3,000 years before the Han period creates archaeological precedent that the Chinese government has a documented interest in containing. The bones are under glass. The research is under an access regime that has not opened. What population, genetically distinct from all known ancient Eurasian groups, occupied a basin in western China in 1800 BCE and maintained complete genetic isolation for centuries? Where did the ancient North Eurasians go? What is the surviving ghost population whose genetic signature sits inside the Tarim Basin mummies and exists in no modern population in pure form? And why does the country with the most complete collection of these individuals restrict the research that would answer these questions? The desert preserved them. The politics are keeping them there. Across the Eurasian continent and out into the Atlantic, another isolated population occupied a string of islands no one can explain how they reached, and the bones they left behind still don't answer it. Number 4. The Guanches. You're on the island of Gran Canaria in 1402. Norman knights Jean de Bethancourt and Gadefort de la Salle, dispatched by the Castilian crown, complete the first European colonization of the Canary Islands and begin documenting the people they find there. The Guanche are tall, light-skinned, and speak a language bearing no relationship to any North African tongue the colonizers know. They have no boats capable of reaching any neighboring coastline and no oral tradition explaining how they arrived. And they have been doing something the arriving Europeans recognize immediately from accounts of ancient Egypt, mummifying their dead in the absence of any documented contact with Egypt, for as long as anyone on the islands can recall. The mainstream explanation is North African Berber origin. The Canary Islands lie roughly 65 miles west of the Moroccan coastline. Neolithic-era North African populations reached the islands by coastal boat during a period of maritime competence they subsequently lost, populated all 7 islands, and developed in isolation for approximately 3,000 years. The mummification is an independent development. The large stature recorded in Spanish accounts reflects observer enthusiasm rather than anthropological measurement. Here's what makes physical anthropologists uncomfortable. The stature is in the bones, not in the accounts. Skeletal analysis conducted by Julio Gonzalez and colleagues at the Universidad de la Laguna across multiple Guanche burial sites documented mean male height ranging from 5 feet 8 inches to over 6 feet across the skeletal sample. The mean European male height in the 15th century was approximately 5 feet 5 inches. The mean stature of contemporary North African populations was lower. The large stature of the Guanche people is not anecdotal observer error. It is in the skeletal record measured by 20th century researchers with no connection to the colonial account. Their bones are taller than anyone's bones who should logically have been their ancestors. The mummification is a more serious problem. Guanche mummification involved evisceration, desiccation packing with botanical materials, and wrapping in animal skin following a prescribed sequence. Francisco Hernandez de Bori, an anatomist who examined Guanche mummies in Tenerife in the 18th century, compared their preparation to Egyptian specimens and found the procedural sequence cavity clearance, vegetable matter packing, prescribed limb positioning, specific burial orientation more similar than different. No trade route or documented maritime contact between Bronze Age Egypt and the Canary Islands has ever been established. 2 cultures separated by 2,000 miles of ocean and 1,500 years developed the same elaborate funerary technology. A 2017 ancient DNA study by Cosimo Post and colleagues at the Max Planck Institute for Evolutionary Anthropology, published in Current Biology, sequenced 14 Guanche individuals from multiple burial sites. The haplogroups confirmed North African Berber origin in the maternal line, consistent with the migration hypothesis. The finding that made the paper unusual is what was absent. No genetic contribution from sub-Saharan Africa, ancient Iberians, Phoenician maritime traders, or any other population active in the western Mediterranean during the 2,000 years the Guanches inhabited the islands. Phoenician, Roman, and Arab traders all operated in these waters. None left a single genetic trace on an island 65 miles from the African coast. 2,000 years of documented maritime traffic through waters adjacent to the Canaries, and the Guanche genome remained a closed system. That is not isolation. That is something more deliberate, or more protected, than isolation normally permits. How did a Neolithic North African population develop mummification techniques structurally parallel to ancient Egypt with no documented mechanism of contact? What produced a skeletal record consistently taller than any neighboring population? And why does the genome of an island people 65 miles from a trafficked coastline show zero detectable genetic input across 2 millennia of documented maritime activity? They disappeared within 2 centuries of European contact. Their bones are in museums across the islands. Their answers have not followed. On the opposite side of the Atlantic, a man drowned in a river and spent 9,000 years becoming a question that science and federal law have not resolved. Number 5. Kennewick Man's people. You're on the bank of the Columbia River near Kennewick, Washington, on July 28, 1996. Will Thomas and David Deasy, attending a hydroplane race, step into shallow water and find what they take for a smooth rock. It is a skull. The Benton County coroner calls archaeologist James Chatters, who spends the following weeks assembling a nearly complete skeleton from the riverbed. Radiocarbon dating of a femur returns 8,410 to 9,410 to 9,400 bow. Before the present among the oldest and most complete human remains ever recovered in the Americas. Then Chatters sits down with the skull, begins taking measurements, and the question that has not been closed since begins. The mainstream explanation is that Kennewick Man represents an early American population whose physical features fell within normal human variation for a 9,000-year-old founding lineage, and whose descendants are the Native American peoples of the Columbia Plateau. Early morphological assessments were influenced by inappropriate modern reference populations. The bones are ancestral Native American remains. The tribes who claimed them were correct. Here's what makes paleoanthropologists uncomfortable. The morphology was real, documented by independent measurement, and remains unexplained by the genetic result. James Chatter's initial assessment noted that the skull's cranial indices placed it outside the expected range for populations derived from northeastern Asian founding groups. Specifically, the skull was dolichocephalic narrow and long from front to back, while most modern Native American and Northeast Asian skulls are broader relative to their length. This is not racial interpretation. It is the same biometric measurement applied to any skull. The finding was confirmed by forensic anthropologist Joseph Powell at the University of New Mexico, who applied multivariate statistical analysis to Kennewick Man's skull measurements against a worldwide reference database. Powell's 1999 analysis placed the cranial measurements outside the range of modern Native American populations, with closest statistical affinity to South Asian and Polynesian reference groups. The legal battle that followed ran for 8 years. The Army Corps of Engineers, in whose jurisdictional waters the bones were found, moved immediately to transfer them to Columbia Plateau tribal claimants under NAGPRA, the Native American Graves Protection and Repatriation Act. A coalition of scientists sued for study access. Federal courts sided with the scientists in 2002. The resulting research was the most comprehensive osteological and contextual study ever conducted on Paleo-American skeletal remains. In 2015, Ancient DNA Analysis by Morten Rasmussen and colleagues published in Nature established Kennewick Man's genetic affiliation with high confidence. He is most closely related to modern Native American populations, specifically the Colville tribe among the Columbia Plateau claimants. The DNA resolved the tribal claim definitively. It did not resolve the morphology. A 9,000-year-old individual genetically ancestral to modern Native Americans does not resemble them by skull measurement. That is not a contradiction in the data. It is the data. The founding population of the Americas carried a skull type that does not match the descendants who inherited their genes. Something changed cranial morphology across an entire continent over 9 millennia. The direction of change from the dolichocephalic Kennewick type toward the broader modern Native American skull implies either replacement by later migrants who did not carry the same morphology, or selective pressure at a rate no current model documents. Kennewick Man is not alone. Luzia woman, recovered from Lapa Vermelha cave in Brazil in 1975 and dated to approximately 11,500 years before present by Walter Neves at the University of São Paulo, shows the same dolichocephalic morphology with the same absence of Northeast Asian cranial features. Two individuals, thousands of miles apart, at the oldest dates available in the Americas, with the same skull type that disappears as the millennia pass. A founding population replaced or absorbed. By whom, over what timeline, and from where none of it is established. Who were the people who carried the Kennewick skull type? Where did they originate before they reached the Americas? What population arrived after them and displaced or absorbed them fully enough to transform cranial morphology across an entire continent? He lay in a river for 9,000 years. The question he represents has been open since the day he was found. 4,000 miles east of where he drowned, a population emerged from the grasslands north of the Caucasus Mountains around 3000 BCE and transformed the genetics of 2 continents in a span of centuries, and no one has fully explained what made them capable of it. Number 6. The Yamnaya. You're in a laboratory at the Max Planck Institute for Evolutionary Anthropology in Leipzig in 2015. Johannes Krauss and his ancient DNA team are completing analysis of skeletal material from Kurgan burial mounds across the Pontic-Caspian steppe, the vast grassland stretching from modern Ukraine east to Kazakhstan. The individuals in these mounds died between 33–2500 BCE. They belong to the archaeological culture designated the Yamnaya, named for the Russian word for pit, after their characteristic pit grave burial style. When Krause's team feeds the Yamnaya genome into the reference database, the result produces something that David Reich at Harvard Medical School, publishing the companion paper the same month, describes as one of the most significant findings in the history of population genetics. The Yamnaya carry a genetic combination that does not clearly match any population alive on Earth. The mainstream explanation is Bronze Age migration. The Yamnaya were nomadic cattle pastoralists who domesticated horses for riding, invented or adopted the wheeled wagon, and exploited the steppe grasslands with a mobility that prior populations lacked. Around 3000 BCE, they expanded rapidly west into Europe and east toward South Asia. Their descendants include the corded ware culture of Central Europe and the Indo-Iranian populations of the Eastern Steppe. The genetic expansion is real and documented. Modern Northern Europeans carry between 50 and 75% Yamnaya ancestry. It is not in question. Here's what makes population geneticists uncomfortable. The replacement was not what migration looks like. Wolfgang Hock, Iosif Lazarides, Nick Patterson, and David Reich, publishing in Nature in February 2015, analyzed ancient DNA from 69 individuals spanning 9,000 years of European prehistory. The data does not show gradual mixing. Between 45–2500 BCE, the genetic ancestry of Northern and Central Europe transforms almost completely. The Neolithic farming populations who had occupied Europe since 7000 BCE, who built the megalithic monuments, the long barrows, Apastamat. The timber halls collapse from near complete genetic dominance to less than 25% of the regional ancestry within a few centuries. The Yamnaya component fills the space. The Y chromosome record is the part that has not been explained. The male lineages of Neolithic Europe haplogroups dominated by G2A, which had been the majority Y chromosome type in farming Europe for thousands of years, disappear from the archaeogenomic record in the affected regions during this transition. Not diluted, not reduced, gone. Only Yamnaya-derived Y chromosome lineages appear in the post-transition record. This is the genetic pattern you get from reproductive exclusion of one male lineage by another, not from peaceful migration and assimilation. What mechanism produced near total replacement of the male lineage while leaving some Neolithic female ancestry in the subsequent population is not established by any published model. The skeletal record carries its own anomalies. Yamnaya individuals averaged 5 feet 10 inches tall, 3 to 4 inches taller than the Neolithic European farmers they replaced, who averaged between 5 feet 5 and 5 feet 7. The diet signature in their bones differs. Heavy reliance on animal protein from a pastoral economy rather than the grain-based diet of European farmers. Their skulls are dolichocephalic, a different morphological type than the broader-skulled Neolithic population. They represent a different body type, a different diet, and a different genetic heritage arriving abruptly at the edge of agricultural Europe. The Yamnaya themselves are a genetic puzzle. Their ancestry derives from eastern hunter-gatherers of the Eurasian steppe, combined with a Caucasian hunter-gatherer component from populations near the Caucasus Mountains, a combination that did not exist in Neolithic Europe, Anatolia, or anywhere in the Levantine farming world, and is not clearly present in any living population in undiluted form. The Yamnaya assembled from two ancestral components that appear almost nowhere in the pre-Yamnaya record, emerged as a dominant force that reshaped two continents and dissolved into the populations they transformed. They have no surviving homeland in any genetic sense. They are everywhere in their descendants and nowhere as themselves. What happened to the Neolithic Europeans? What mechanism explains near-total Y chromosome replacement while preserving some female ancestry? What were the Yamnaya before the steppe, and where did the genetic combination that made them come together? And how did a population that did not exist as an identifiable group before 3500 Ananacoint, before 3500 BCE, come to carry the ancestry of half of humanity's largest continent within 500 years? 1,000 miles east of their homeland, in a cave in the Altai Mountains, something much older left a single fingertip bone in the sediment, and what it contained rewrote everything. Number 7. The Denisovans. You're in Denisova Cave in the Altai Mountains of southern Siberia in 2008. Russian archaeologists have been working this site for decades. The cave's stratigraphy spans hundreds of thousands of years, with Neanderthal bone fragments and modern human artifacts occupying separate layered deposits. In the cave's third cultural layer, a team member finds a bone fragment roughly the size of a child's fingertip. It is the distal phalanx of a juvenile finger from a child estimated between 5 and 13 years old. The fragment is sent to Svante Pabo's team at the Max Planck Institute for Evolutionary Anthropology in Leipzig, specialists in recovering genetic material from degraded ancient bone. In 2010, Pabo's team publishes their results in the journal Nature. The DNA extracted from the finger bone does not belong to a Neanderthal. It does not belong to a modern human. It belongs to a previously unknown species of hominin, an entire branch of the human family tree that science had never identified before, established from a bone smaller than a thumbnail. The mainstream scientific response is enthusiasm. A new species, ancient, extinct, recovered through the triumph of paleogenetics. The find is celebrated. What the public discussion moves past quickly is the implication sitting at the center of the data. The Denisovans did not simply coexist with modern humans and then vanish. They interbred with them. Population genetics research conducted by David Reich at Harvard Medical School, published beginning in 2010, established that populations from Melanesia, Aboriginal Australia, and parts of Southeast Asia carry between 4 and 6% Denisovan DNA in their genomes. These are not signals at the edge of detection. They are substantial, consistent genetic contributions from a species that no one had identified before 2010, present in the living bodies of tens of millions of people. Those people have been carrying Denisovan genes, using them, passing them to their children, without knowing what they were or where they came from. The biological significance of that inheritance is not abstract. Tibetan people carry a specific variant of the gene EPase1, which governs the body's response to low oxygen concentration at high altitude. Without this adaptation, humans living above 14,000 feet develop chronic mountain sickness, suffer reduced fertility, and experience shortened lifespan. Tibetans do not experience these effects. The specific EPase1 variant they carry has been traced by Emilia Horta-Sanchez and colleagues in a 2014 study published in Nature to Denisovan ancestry. Tibetan people can live where they live because a now extinct species, known from a cave in Siberia by a single finger bone, gave them the genetic means to do it. The Denisovans are not ancient history. They are functional, operating biology inside people alive today. What the Denisovans looked like remains unconfirmed. The archaeological record for the entire species consists of several bone fragments, a few teeth, and one skull fragment found in 2019 at Denisova Cave. The 2019 specimen comes from the daughter of a Denisovan father and a Neanderthal mother, the first direct physical evidence of reproduction between these two species preserved in a single bone. In 2021, researcher Mayan Harel at the Hebrew University of Jerusalem extrapolated a facial reconstruction from methylation patterns in the Denisovan genome. The image suggests a being with a wider skull, a wider and more projecting face, and a brow ridge architecture distinct from both Neanderthals and modern humans. We have the genome. We have living human populations carrying their genes in functional form. We have biological traits in modern people that trace directly to a Denisovan inheritance. We do not have the face. How many species of humans existed simultaneously in the period when Denisovans were alive? If Denisovans interbred with modern human ancestors across multiple geographic populations, how many other unknown species are hidden inside the modern genome, waiting for another finger bone to name them? What else did we inherit? And why does the species that gave Tibetan people the ability to breathe at altitude leave only a fingertip and a handful of teeth as evidence of its entire existence on Earth? We carry them inside us. We still don't know what they were. In Indonesia, on an island where giant lizards still walk, another species of human survived until almost within human memory, and the bones they left behind make no sense by any current model. Number 8. The Flores hobbit. You're in Liang Bua Cave on the island of Flores in Indonesia in 2003. Australian archaeologist Mike Morwood and Indonesian colleague Thomas Sutikna are working through sediment layers that have already yielded stone tools of unexpected sophistication for the age of the deposit. In a layer dated to approximately 70,000 years ago, they find a partial skeleton. Adult female, based on pelvic morphology. The skull is intact. They begin measuring. The skeleton is 3 feet 7 inches tall. The brain case contains between 380 and 420 cubic centimeters of volume. A chimpanzee's brain averages 400 cubic centimeters. This is smaller than any known specimen of Homo erectus, smaller than Homo habilis. The tools found nearby require planning sequences too cognitively complex to associate with that brain volume by any established model. They designate the specimen LB1, the species, Homo floresiensis. The popular press calls it the hobbit. The scientific community begins an argument that has not stopped. The mainstream explanation is island dwarfism. Large mammals isolated on islands with limited resources evolve smaller body sizes through a process called insular dwarfism. Flores is isolated. Homo erectus was present in the surrounding region. The lineage scaled down to what LB1 represents. This is established evolutionary biology. The dwarf elephants of Sardinia and the dwarf hippopotamus of Cyprus follow the same logic. Here's what makes evolutionary biologists uncomfortable. Island dwarfism does not do this to brains. Documented insular dwarfism in mammals produces body size reduction, with a proportional but less severe reduction in brain volume, following known allometric scaling relationships. A dwarfed lineage gets a smaller version of its ancestor's brain. Not a brain smaller than the most archaic known member of its genus. To get from Homo erectus's brain volume to LB1's 380 cubic centimeters through resource limitation alone violates the known scaling rules. You cannot shrink a Homo erectus brain this far by limiting its diet on an island. The math produces a different result. A 2016 analysis by researcher Mana Dembo at Simon Fraser University, published in the Journal of Human Evolution, conducted a systematic comparison of LB1's skull morphology against every known hominin species. Dembo's analysis found that LB1's skull architecture is not consistent with a dwarfed Homo erectus lineage. The morphological affinities place the Flores specimen closer to earlier lineages, possibly Homo habilis or something more archaic still. If Dembo's analysis is correct, the species that became Homo floresiensis was not a shrunken Homo erectus. It was something older that reached Flores by a route and at a time we have not established. The tools complicate everything further. The stone implements found in association with LB1 require multi-stage production sequences. Selecting the right raw material, shaping a blank, removing flakes in a specific order to produce a functional edge. Cognitive archaeologists associate this production complexity with planning ability and abstract problem-solving capabilities. The brain generating those tools, at 380 to 420 cubic centimeters, is operating at a level that no established neuroscientific model can account for at that volume. Either the tools didn't belong to LB1, or something about the relationship between brain size and cognition is not what we think it is. The people of Flores have a name for them. Ibugogo means, roughly, grandmother who eats anything. The folklore describes small, hairy, bipedal beings who lived in caves, stole food from villages, had their own language, and could repeat human words after hearing them once. In some accounts, the last Ebu Gogo were reported in the 19th century, within the period of colonial written records, centuries after the fossil record places Homo floresiensis's extinction at approximately 50,000 years ago. Whether those accounts represent a memory carried across 50 millennia or a more recent encounter is a question no field archaeologist has yet closed. The cave at Liangbua is still being excavated. They keep finding things. In Florida, in 1982, a construction worker found bones in a pond, 8,000 years old, with the brains still inside. And the DNA in those brains refused to match anyone expected. Number 9. The Malta skulls. You're on the island of Malta in 1902. Construction workers breaking ground in the Paula district strike something hollow beneath their feet. What opens under their tools is a subterranean complex carved into the living limestone over 3,000 years before the Roman Empire, before classical Greece, before any civilization the textbooks call foundational. They name it the Hal Saflioni Hypogeum. Archaeologist Themistocles Zamet begins excavation in 1905 and uncovers approximately 7,000 individuals, the largest prehistoric ossuary ever found. Its oldest burials dated to 3600 BCE. Some of the skulls among them do not look like anyone who should be buried in Neolithic Europe at that time. The mainstream position on the hypogeum is that it represents the Neolithic farming population that settled Malta from Sicily approximately 5,500 years ago. The human remains are within normal morphological variation for that founding population. The site is a UNESCO World Heritage location, thoroughly documented, carefully maintained. The standard academic literature does not treat the skeletal material as anomalous. Here's what makes physical anthropologists uncomfortable when they examine the full sample. The unusual skulls are not minor variations. Among the skeletal material recovered, a subset shows significant elongation of the cranial vault extending backward from the crown. There are no associated trauma markers. There is no evidence of binding devices or cloth in the burial context. There is no documented parallel for deliberate cranial deformation in any Neolithic, European, or North African population from which Malta's founders are understood to have derived. Maltese physician and researcher Dr. Anton Mifsud documented these specimens in published work from the 1990s, noting that the elongated examples consistently display an absence of the sagittal suture, the joint running along the top of the skull between the two parietal bones that appears in every modern human skull without exception. This is not the first time in this video you have encountered that absence. The Paracas skulls in Peru show the same consistent structural anomaly. No sagittal suture, one continuous plate of bone where two joined pieces should appear. 5,000 miles of ocean separate these two populations. More than 1,000 years separate their periods of burial activity. No documented contact between pre-Columbian Peru and Bronze Age Malta has ever been established. Either this is a coincidence statistical tables would not predict, or the same morphological type was present on both sides of the Atlantic for reasons no current model accounts for. The DNA situation at Malta is more opaque than at Paracas. A sample submitted for genetic analysis from hypogeum skeletal material in the early 2000s returned results that, according to the researchers who commissioned the test, were not published. The reason given was sample degradation producing inconclusive results. Sample degradation in Mediterranean limestone is a genuine and documented preservation problem. It is also the explanation you would give if the results returned something the available interpretive frameworks were not prepared to accommodate. The broader Neolithic Maltese genome is better established through other sites. A 2019 genomic study led by Wolfgang Hock, published in Nature, confirmed that the temple-building population of Malta derived primarily from Neolithic Anatolian farmers, consistent with the southeastern European agricultural expansion model. That study did not specifically target the elongated skull specimens from the hypogeum. The unusual skulls remain unsampled in the published scientific literature. The 7,000 individuals buried here were displaced from their original positions across multiple excavation phases. Precise documentation of which skulls came from which location within the complex is incomplete. The elongated specimens, once displayed publicly at the National Museum of Archaeology in Valletta, are now held in storage, accessible by institutional request. The casts are on display. The originals are in the back. What population brought skulls with no sagittal suture to Malta in 3600 BCE? Why does the same structural feature appear in Paracas among a people with no documented connection to anything in the Mediterranean? What did the early 2000s DNA sample actually return? And why do the originals sit in storage while the replicas greet the visitors? 7,000 people were buried here. A few of them were different. No one has officially explained how. The Malta skulls bring us full circle, back across the Atlantic, to a construction pond in Florida, where the oldest preserved human brains on Earth were found, and the DNA inside them refused to behave. Number 10. The Windover Bog People. You're at a construction site in Titusville, Florida in 1982. A backhoe operator named Steve Vanderjagd is draining a pond to make room for a drained housing subdivision when he notices dark shapes in the muck at the pond's bottom. He stops the machine. The shapes are skulls. The pond is a peat bog, and what it has been doing in the dark for the past 8,000 years is preserving things. Archaeologist Jim Adavasio from the University of Pittsburgh is brought in. What his team excavates over the following field seasons will become the most significant Paleo-Indian skeletal discovery in North American history and the most uncomfortable one. The Bob has preserved 168 individuals, radiocarbon dated to between 7,000 and 8,000 years before the present. That places them among the oldest intact human remains in the Western Hemisphere. But the peat did something no other Paleo-Indian site has replicated. Its acidic, anaerobic chemistry, airless and dark, preserved not only bone but brain tissue. 91 of the skulls contain original brain matter, still present, still recoverable, still carrying extractable genetic information 8,000 years after burial. No other site on Earth has produced anything comparable in volume or preservation quality. The mainstream interpretation is straightforward. an ancient Paleo-Indian population using a pond as a cemetery over an extended period. The individuals show normal morphological variation. Analysis has proceeded carefully over several decades. Here's what makes population geneticists uncomfortable. The DNA does not match the expected model. The founding of the Americas is understood to derive from northeastern Asian populations, crossing via the Bering Land Bridge between 15,000 and 20,000 years ago. Mitochondrial haplogroups A, B, C, and D dominate Native American populations and trace directly to northeastern Asian origins. This is the baseline. The Windover people carry haplogroup X2A. Haplogroup X is present in certain Native American populations, notably the Ojibwe of the Great Lakes, where it appears at frequencies reaching 25%. It is entirely absent from northeastern Asian populations. It appears in Middle Eastern populations, and in specific European lineages not in Siberia, not along any known Asian migration corridor. The Windover population are among the earliest confirmed carriers of haplogroup X in the Western Hemisphere, appearing in a Florida bog at an age that predates any European arrival by several thousand years. Dennis O'Rourke at the University of Kansas, one of the principal analysts of ancient Native American genetics, noted in published work that haplogroup X's distribution in the Americas is not consistent with a simple single-entry model from Northeast Asia. What is consistent with it is a separate entry from a separate direction at a separate time. Dennis Stanford of the Smithsonian Institution and Bruce Bradley of the University of Exeter proposed in their 2012 Solutrean hypothesis that haplogroup X entered the Americas via transatlantic contact, with a population from Europe or the North Atlantic moving along the edge of the Ice Age ice shelf and reaching the eastern seaboard. A second founding population, separate from the Bering migrants, arriving by a route the textbooks do not include. Stanford and Bradley's argument remains contested. It has not been disproven. The brains of the Windover people are in storage at the Florida Museum of Natural History. 91 sets of preserved neural tissue, 8,000 years old. Modern genomic sequencing has advanced dramatically since the initial analyses. A comprehensive study using current technology applied to the full Windover tissue collection would either resolve the haplogroup X question or reveal that the question is larger than anyone has publicly stated. That study has not been funded. The bones don't match the model. The brains contain the answers. The study hasn't been done. These are what 10 sets of bones add up to. A royal Egyptian family whose identical skull anomaly vanished when the dynasty was erased, 429 Peruvian crania with 60% more internal volume than any bound skull and DNA, matching nothing on record, Bronze Age mummies in a Chinese desert descended from a ghost lineage nowhere in the living world, an island people 65 miles from Africa whose genome absorbed nothing from 2,000 years of maritime traffic, a 9,000-year-old American whose skull does not match the descendants who carry his genes, a steppe population that replaced every Neolithic European Y chromosome in a few centuries and then dissolved, an entire species from a finger bone whose genes let Tibetans breathe at altitude today, 3'7" toolmakers in Indonesia whose descendants reportedly saw them in the 19th century, elongated Malta skulls missing the same bone absent in skulls across the Atlantic, and 8,000-year-old brains in a Florida pond carrying DNA the standard model cannot place. The pattern keeps surfacing in bones, because bones are the one thing that does not disappear fast enough to be cleanly denied. Subscribe so you don't miss the next one. Like if you think the official prehistory of our species has more gaps than any textbook admits. And drop a comment telling us where in the world you're watching from.